So good afternoon, everyone. Welcome to the Pacific Salmon Commission Seminar Series on Environmental Variation and Pacific Salmon Management, the view from both sides of the border. You should be able to see the seminar series flyer on your screen. My name is John Holmes, and I'm the Canadian co-chair of the Coho Technical Committee, and I'm pleased to introduce this seventh seminar in our monthly series titled The Effects of Climate on Marine Salmon Productivity, featuring Dr. Jan Ohlberger from the Science Division of Washington Department of Fish and Wildlife and Dr. Brendan Connors from the Stock Assessment Division of Fisheries and Oceans Canada. So our seminar today is on salmon productivity and how it's impacted by climate competition. Many of our models and management processes implicitly or explicitly assume that there's stationarity in productivity. And while that might be true for short periods of time in specific area, over the periods of time in which many of us have worked with salmon, we know that salmon productivity is changing in response to both ecological and environmental conditions, particularly climate. We need to find ways of incorporating this temporal variability into our conservation and management approaches so that decisions are informed by this changing context. Our speakers today will provide insights into the changing dynamics of climate and competition on salmon productivity. Now, before we go any further, let me introduce our speakers. Brendan Connors is a research scientist with Fisheries and Oceans Canada, based out of the Institute of Ocean Sciences on southern Vancouver Island. His background is in salmon population ecology, and his research program at DFO is focused on quantitative methods to understand salmon responses to a changing world, biocomplexity, and mixed stock fisheries in large river basins, and fishery risk assessment. Brendan recently joined the PSC family as the Canadian co-chair of the Committee for Scientific Cooperation, and he also co-chairs the Pacific Working Group for DFO National Marine Fisheries Service Climate and Fisheries Collaborative Initiative. Jan Ohlberger is a research scientist in the Science Division of the Fish Program at the Washington Department of Fish and Wildlife. He's broadly interested in understanding how fish population dynamics, demography, and life history change in response to altered ecological and environmental conditions, and how this affects their conservation and management. His research is focused on salmonids, and he's currently developing management strategy evaluations for salmon and steelhead to identify harvest strategies that balance biological and socioeconomic objectives. Before joining WDFW, he was a research scientist in the School of Aquatic and Fisheries Sciences at the University of Washington and a postdoctoral fellow at the University of Oslo in Norway. He obtained his PhD at Humboldt University of Berlin in Germany. So the plan for today is to hear about Pacific salmon responses to a warming, more crowded ocean from Brendan, followed by the effects of climate and competition on the productivity and demography of Pacific salmon from Jan. And Jan will also provide a slide at the end of his presentation to see the Q&A session that will follow. So after the presentations concluded, there'll be a short break followed by a moderated question and answer session. Those of you that are watching this will know that you cannot unmute your microphone or turn on your video, but you can use the Q&A feature in the controls at the bottom of your screen. So if you have a question, please put it into the Q&A. And if there are enough upvotes, it will be asked by our moderators. Everyone can upvote the questions posed in the Q&A feature, and the moderators are going to choose the highest voting questions posed to our speakers. If there are more questions than can be answered during the time available, we will endeavor to get written responses from our speakers and include those responses in the seminar package. The seminar package will include a recording of this seminar, flyer and other materials, plus any written responses. And so thanks, and now on with the presentations, beginning with Brendan. Take it away, Brendan.
Good afternoon, everyone, and thanks a lot, John, for the introduction, and to the rest of the organizers of the seminar series for the invite. I'm really thrilled to have the opportunity to share some perspectives on the effects of climate on Pacific salmon productivity, which for the purposes of this first presentation, I'm kind of reframing as Pacific salmon responses to a warming and more crowded ocean. Jan and I have tried to coordinate a little bit on our presentations, and I'm grateful that he's letting me go first since he's a pretty tough act to follow. And as John mentioned, we have pulled together a slide that summarizes some key questions to seed discussion at the end of the presentations. So before I get started, I do want to acknowledge my co-authors on those bits and pieces of work that I present today that I was explicitly involved in. I'm drawing on quite a bit of other work, but I did want to put a shout out to those that contributed to the work that I've specifically involved in, as well as those organizations and bodies that have helped contribute some of the funding to the work that I'll be talking about today. So as I think everyone is probably acutely aware, the North Pacific is getting warmer, both on its surface and at depth. It's experiencing more extremes like marine heat waves. And that's illustrated here on this slide, just on the left at the North Pacific scale, with both annual and 3-year running average sea surface temperature anomalies over the past century and a half or so. The key take home here from this slide on the left is you can see we are now way outside the envelope of historical variability, at least in this case in sea surface temperature over this period of time. Regionally, warming's occurring faster in more northerly latitudes than in southern ones. That's kind of consistent with what we'd expect with Arctic amplification of global warming. And that's illustrated here on the right with sea surface temperature in the eastern Bering Sea, the top right panel, and contrasted with sea surface temperature in coastal British Columbia and the bottom panel. You can see that particularly at the tail end of that time series, the rate of warming and magnitude of warming is certainly larger up in the Bering Sea. What you can also see if you really squint in at the very end of these time series is the beginnings of a fairly recent dip back down in SST anomalies after the back-to-back marine heat waves we've had in recent years, associated with the now what are maybe will be a triple dip La Nina, which may have been responsible, at least in part, for some of the better than perhaps expected returns that we've seen in some salmon systems along the coast this past year. Just a hypothesis, but throwing it out there. This warming in the North Pacific can now be unequivocally linked to us, and that is anthropogenic climate change. Illustrating that here with a climate attribution statistic called the fraction of attributable risk, or FAR, which is plotted out here from the mid-1950s through to present on the right-hand side. And FAR indicates the probability that a given set of observations, in this case sea surface temperature, are attributable to anthropogenic forcing. We can calculate it using hindcasts of global climate models, and a FAR value of 0 indicates that are observations that are equally likely with and without human influence. And a FAR value of 1, so up at the top end here, indicates observations that are essentially statistically impossible without anthropogenic forcing or human influence. And so what this plot is meant to illustrate and highlight is that at the North Pacific scale, sea surface temperature has now effectively become fixed in an anthropogenic state. Now, you might be forgiven for thinking, or you might think, this is probably not a good thing for Pacific salmon, but there are now, on average, more salmon in the North Pacific than there have been at any time in the past century. And that's illustrated here with a plot of total North Pacific returning abundances of pink, chum, and sockeye salmon from the mid-1920s through to present. Pink, chum, and sockeye make up the lion's share of the abundance in biomass. So coho and chinook, don't want to forget them, are not in this time series. What this time series also highlights, if you look over at the far right-hand side, is that 2018, 2019, and 2021 saw the 3 highest return abundances of these 3 species combined over this period of record. Now, while the majority of salmon produced across the North Pacific are of natural origin, an increasing fraction, now about 20% of total pink, chum, and sockeye production is from hatchery-produced fish. These salmon overlap to varying degrees at sea, and they're hypothesized to compete both directly and indirectly for sea while they're at sea. Indirectly for food while at sea, while also having broader ecosystem effects on everything from plankton to other fishes to birds and even possibly marine mammals. Now, there's a pretty solid and substantial body of evidence that Pacific salmon survival is influenced by a warming ocean. And there's growing body of evidence that increasing abundances of Pacific salmon, particularly pink salmon, but not exclusively, are associated with reduced survival in other salmon species. But these relationships vary in space. And one illustration of this is from some work that we did a couple of years ago, looking at sockeye salmon responses to a warming and more crowded ocean across their northeastern Pacific range. So this spans populations from the Fraser, which are highlighted here in populations 1 to 19, all the way up the coast, all the way into Bristol Bay and just north, which would be these ones entering into the Bering Sea. What this plot on the left shows, if we just start with the top panel, is the posterior distributions of the predicted percent change in recruits produced per spawner for individual populations, those are those thin lines, or for the kind of overall hyper global effect within regions, those are the thick lines, for a given magnitude of warming, increase in sea surface temperature, in this case, a standard deviation unit increase. And what we see, again, if we focus on this top panel up here, is that responses to warming vary very strongly latitudinally. So at the southern end of their range, they're negative, and as you move north, they become positive, and even more positive once you work your way up into the Bering Sea and sockeye populations like those in Bristol Bay, where the positive response is about twice as large as it is for those two other regions. If we move down one panel, now what we have here is the predicted change in recruits produced per spawner for an increase in potential competitors. In this case, the abundance of North Pacific wide pink salmon in the second year of sockeye marine life. And what we see here is consistent direction of responses, but varying magnitudes, with the magnitude being larger for those at the more southern end of their range. And then lastly, on the bottom panel here, we have the combined effects of increases in both sea surface temperature and competition, where warming temperatures appear to offset the negative effects of competition in the north, particularly up here in the Bering Sea, but appear to exacerbate them further down south, down here in the green. Now, not only do salmon responses to a warming and more crowded ocean vary in space, like I just highlighted in that last slide, but there's also pretty substantial now body of evidence that at least for warming, they also vary within regions over time. And that's illustrated here for just a subsection of populations from the Gulf of Alaska, pink, sockeye, and chum populations, and their relationships or responses to increases in SST, in this case, SST in the winter preceding marine entry. And what you can see, if we just focus above this dashed line to begin, these are the correlations between productivity and sea surface temperature in the years prior to the 1988-89 ocean marine regime shift. And then below the line are responses relationships after the regime shift. And what you can see is that there are consistently positive, though variable magnitude relationships for both pink, sockeye, and chum prior to 1988. And those relationships all but disappear after 1988. I believe these time series ran up until the early 2010s. So not only do responses vary in space, they almost certainly also vary through time. And we see evidence for not just changes in productivity associated with warming and with competition, but also changes in other life history characteristics like age at maturity or things like growth. So here I have an illustration of relationships between Fraser River sockeye salmon age at maturity, or the size of spawners on the spawning grounds, and North Pacific wide pink salmon abundance in the second year of sockeye marine life. So on the left here, this is the change in log odds of maturing at 5 years of age instead of 4 years of age as pink salmon abundance increases. And then on the right is the change in fork length of spawners as pink salmon abundance increases. There's a lot going on in these plots, but the important thing to focus in on is this red line, which is the kind of overarching global relationship across the populations. And then the individual black dots are population level responses. And what you can see is that as a competitor abundance increases, an index of competitor abundance increases, fish tend to mature later. So they're further along this axis here, and they tend to return at a smaller size. But those relationships vary amongst populations. They're all generally in the same direction and of relatively similar magnitude. In addition to affecting survival and growth, ocean warming is also expected to reduce the amount of suitable thermal habitat for Pacific salmon across the North Pacific. So this figure here, which I've taken the liberty of recreating for the purposes of this presentation, not so well, but good enough for today, illustrates a projected 50% reduction in summer thermal habitat for sockeye from the 1980s through the 2080s under a high emission scenario back when the work was done. And then the table below summarizes the projected percent change in summer thermal habitat for other species over the same time period. And what it really highlights is that those species with the narrowest thermal tolerance, like sockeye and Chinook, are projected to be the most sensitive to thermal habitat change under global climate change. These general patterns in terms of changing and shrinking thermal habitat in the ocean have been shown by a number of others as well. And what they suggest is that shrinking ocean habitat due to warming may further exacerbate competition amongst salmon at sea. A warming ocean is not only expected to reduce thermal habitat, it's also expected to result in changes of the way in which salmon are distributed at sea. And this is nicely illustrated in a relatively recent publication by Ole Shelton and colleagues that used a spatiotemporal population dynamics model that was fit to coded wire tag data for Chinook salmon to estimate the distribution of fall run Chinook over the past 40 odd years, as well as the relationship between distribution and sea surface temperature and projected distribution of Chinook, these focal stocks under climate change. So on the left, if we focus in there first, is the distribution of Snake River Chinook salmon among 14 coastal regions from south at the bottom up to north. Mont, I believe, is Monterey Bay, and NSEAK is North Southeast Alaska. And what they show is the distribution, a proportional distribution in warm, which is in red, cool, which is in blue, and average, which is in purple years, and their projected distribution in 2030 and 2070 under continued warming, which are the gray bars with different magnitudes of ticks between them. What we see is that the distribution of Snake River Chinook is sensitive to ocean temperature. So you can see that there are ups and downs depending on whether it's a warm year or a cool year. And so their proportional distribution is expected to shift north from predominantly off the Washington coast and Southwest Vancouver Island today, or in recent years, to predominantly off Southeast Alaska in the future, which is expected to see, I think, about a 5-fold increase in the proportional occurrence of Snake River Chinook by 2070. On the right-hand side, also taken from this paper, is the cumulative abundance or change in cumulative abundance due to climate-driven distributional shifts for 6 focal southern stocks in 2050 relative to the average conditions over the past 3 decades. And what this highlights here is spatially varying changes, with some areas like Central California, which might be a bit of a surprise, and Northern BC seeing increasing abundance, while others like Northern California and Washington coast seeing declines. An important kind of asterisk or caveat with the projected changes in abundance on the right is that these projections assume that there's no change in the relative abundance or production of Chinook from each of these systems, which we expect to also be influenced as the climate changes, but it was a necessary simplifying assumption for these analyses. That nonetheless really kind of highlight how we might expect warming to shift the distribution of salmon, in this case Chinook, at sea. So because of spatially varying responses to warming, a warming ocean will, and certainly almost certainly already has, shifted production of salmon north, creating new and more opportunities for harvest there, while opportunities for harvest to the south have declined and will likely continue to decline. We see this general pattern in commercial landings, in this case of all Pacific salmon species by country over the past century, which is what's plotted up here on the slide. And what we can see is that it's steadily increased in northern latitudes since the mid-1970s and has generally tended to decline, in some cases quite precipitously, at southern ones. Now, before you get too bent out of shape, of course, there are many other things that are going on that are going to influence commercial landings and harvest, including myriad other stressors, many of which you've heard in the seminar series, that may be affecting salmon and their production, particularly at the southern end of the range. But this general pattern, I'd argue, likely reflects, at least partially, climate-mediated shifts in production at very broad scales. And in recent decades, we've seen very sharp increases in the occurrence and harvest of salmon like chum, for example, up in the Arctic and the Mackenzie Basin, which is highlighted up here in the top right. And then not shown on this slide is that in other parts of the far north, like the Barents Sea, which is in northern Scandinavia, and which is warming about 7 times the rate of the rest of the world's ocean, there's been an absolute explosion of pink salmon in recent years, leading to concerns about ecological consequences and calls for deploying new fisheries to take advantage of those fish. Some of what we see in terms of increasing commercial harvest in recent decades, particularly in the north, is probably due, at least in part, to industrial scale hatchery production that occurs there to support commercial fisheries, right? And that's almost certainly benefited in general from a warming ocean and the general positive effects of a warming ocean at more northern latitudes. What's plotted out here on this slide are trends in total hatchery releases over time by region. Note the scales vary considerably depending on which region you're talking about, the y-axis scales, where both Russia and Alaska have seen steady and ongoing increases in hatchery releases in recent decades, while places like Canada and Japan and the lower 48 have generally seen declines since the 1980s. Of course, like commercial harvest, these trends are going to be confounded with all sorts of other things that are going on that are going to influence releases of juvenile hatchery fish. But I'd argue that while increasing hatchery production in the north can take advantage of generally favorable ocean conditions, at least for now, it may come at the cost of exacerbating conservation risks for salmon populations at more southern latitudes, as those fish that originate from there have to both contend with the negative effects of a warming ocean at the more southern region during early marine life, as well as potentially shrinking thermal habitat and increasing competition for limited resources within it later in marine life. This general kind of idea is illustrated here with some simple analyses that build on the example I gave earlier about sockeye across their range, where what we did is we took the estimated relationships that I showed earlier between productivity and either sea surface temperature or competitors, and asked what's the predicted percent change in recruits produced per spawner on average within each of these regions that would be associated with recent North Pacific-wide hatchery pink salmon production, when you either do, which is in yellow, or don't, which is in purple, also take into account recent marine temperatures. A bit of an asterisk when I say recent, these analyses were only up until about the 2015 brood year, 15 return year, so they're a little out of date. But what these simple analyses highlight and what they suggest is that total hatchery production of pink salmon has likely reduced or contributed to reduced sockeye productivity at the southern end of their range in the magnitude of around 15% on average. But in contrast, above average sea surface temperature conditions in the Gulf of Alaska, and particularly in the Bering Sea over that same time period, are estimated to have either partially, which is highlighted here, or completely offset the negative effects of hatchery production on sockeye productivity in those regions. Of course, I'm taking liberties with ascribing causation here, and I want to, be very clear and careful about not overstepping bounds. Right? These are all correlative analyses, but I think they do help illustrate and put these types of magnitudes of effect in general perspective. And of course, they assume that wild and hatchery fish interact in the same way and are similarly distributed at sea. So there's a number of caveats to it, but nonetheless, it helps put some of those things into perspective. Okay, just about wrapped up here, second to last slide. I wanted to end with highlighting that it's fairly common to throw one's hands up in the air and say, we really can't do anything about what happens to salmon in the ocean. We need to focus our efforts elsewhere in environments where we're more likely to have a positive and real impact on survival, abundance, etc. But based on what I presented here today, I think there's two places where I think we do have some control over conditions that salmon experience at sea. The first is, of course, greenhouse gas mitigation, right? The panel on the left here plots out the year, that's on the y-axis, that North Pacific sea surface temperature is expected to exceed different warming thresholds. Those are along the x-axis, relative to the 1880 to 1950 average for either observed, which is the ERSST, it's the orangest dots, or two emission scenario, moderate mitigation or a business as usual scenario. And what this highlights is that 1 degree of warming is baked in. We're already there. But even with just moderate mitigation, we can in theory significantly delay the rate at which warming occurs and thresholds are exceeded. Right? So addressing global climate change and mitigating the worst of ocean warming, I'd argue, though increasingly difficult and requiring collective action, is not outside of our control. And I think this helps put that in perspective. The second area where we have control is when it comes to things like large-scale, industrial-scale hatchery production. The plot on the right is of total hatchery salmon releases. So I took that previous slide and just combined them all into one over the past century. And it highlights that in recent decades, about 5 billion salmon are released into the ocean each year. And in light of some of the examples that I've provided here today, and I think Jan will build on some of those, this really begs the question of whether or not salmon nations collectively should be considering cooperatively managing and possibly limiting the extent to which hatchery-produced fish are put into the sea. Right? This is not a new idea, although I'll admit it's a bit controversial. It's been suggested numerous times in the past, but the evidence for inter and intra-specific competition at sea, in my opinion, has really rapidly accumulated in recent years. And so in my opinion, provides a stronger foundation of information upon which to ponder this question. Okay, so to wrap up, I want to leave you with these 6 take-home messages from this first part of the pair of presentations. The first is that the North Pacific is getting warmer, thanks to us. It's also getting more crowded, in part also thanks to us. Second, salmon responses to warming vary latitudinally, and their effects are generally negative in the south and generally positive in the north, at least for now. But these responses can and do vary over time and are of course filtered by the habitats that salmon use throughout their life cycle. I didn't dwell on that or highlight that in this presentation, but there's a lot of heterogeneity and a lot of variability that in part is due to the way in which habitat filters global scale climate change. Third, salmon responses to a more crowded ocean include reduced survival and growth and delayed maturation. Fourth, warming will likely reduce both the amount of suitable habitat in the north, shifting it, suitable habitat in the North Pacific, shifting it north, and also redistributing salmon at sea. Fifth, industrial scale hatchery production can take advantage of a warming ocean in the north, but may exacerbate conservation risks in the south. And then sixth, and lastly, what happens in the ocean is not entirely outside of our control. And so management levers that could, in theory, be pulled to help influence the experiences that salmon have at sea should be considered alongside the suite of other levers that are commonly thought of to support salmon recovery and resilience in a warming and more crowded ocean. Thank you. This is the part of the meeting where I wonder if people could hear me the whole time, but I see you, John, so hopefully you could.
Yeah, we could hear you. Thanks very much for the presentation. It was really quite intriguing. And so we'll move on to Jan's presentation.
Yeah, that was a great presentation, Brendan. Thanks. Also wanted to thank the organizers and John for the introduction, and welcome everyone. It's a pleasure to be here and to have the opportunity to talk to you about effects of climate and competition on the productivity and demography of Pacific salmon. As John introduced, so my name is Jan Ohlberger, and I'm currently with the Washington Department of Fish and Wildlife. I put a second affiliation on here, which is the School of Aquatic and Fishery Sciences at the University of Washington, because that's where I did a lot of the work that I'm going to present today. And we've already heard a really great introduction into a lot of the topics: climate change, salmon abundances in the ocean, hatchery production, et cetera. And I'll try to build on that background and some of the topics that Brendan touched on as well, specifically with a few case studies from sort of throughout the West Coast of North America, and with a little more focus on population demography in addition to productivity, and maybe talking a little bit more about some of the mechanisms. Before I dive into the topics, I just also wanted to acknowledge my collaborators and funding organizations that made all this possible. Clearly, the work I'm going to present is the result of many fruitful and fun collaborations over the past few years. So there are many causes of changing productivity and demography in salmon, both natural and anthropogenic stressors that affect growth and survival throughout the salmon life cycle, and that's illustrated here. And I'm showing this to acknowledge that while we're focusing on the effects of climate and competition in the ocean, other factors matter as well that we're not addressing here today. And as indicated, competition and climate change act in both the freshwater and the ocean environment. So focusing in on the topic of today, there are, of course, also multiple mechanisms through which climate and competition affect salmon. And I just want to point out a few important or relevant ones on this slide. So thinking about climate change, for instance, changes in climate can cause changes in frequency, magnitude of floods that cause egg scour. Changes in river flow affect juvenile survival. Freshwater temperatures affect juvenile growth and outmigration timing. And temperatures in the ocean affect growth and survival as well. And then finally, changes in the frequency or magnitude of low flows and heat stress when adults return can cause pre-spawn mortality. And similarly, thinking about competition, which can act both on the intra-specific and interspecific levels within and among species. Now, starting here on the right, competition for juvenile prey and habitat is typically an intraspecific process. We have competition with hatchery origin fish, maybe in river, certainly in nearshore marine habitats, also in the open ocean, where we have competition for food, both as an intra and interspecific process, and then competition for spawning sites, typically within species. I'll touch on most of these, maybe except for the sort of late spawner and the egg stages. I also wanted to provide some motivation for why I am particularly interested in population demography. So the age and size, structure, also sex composition of the populations. The main reason is that population demographic structure integrates the effects or impacts of drivers like climate and competition through changes in growth and survival. So you can think of the demographic structure of a population as a link between individual traits or physiology and population dynamics or productivity on the other side. And as I mentioned, competition affects individuals both through density-dependent processes within and among species, and then climate has direct effects on individual physiology, affecting growth and survival, as well as potential indirect effects through interacting species. Okay, so with that in mind, I want to dig into a few case studies, and I'll start with effects of climate and competition on demography. I'll focus on Bristol Bay sockeye salmon, mostly because I've worked on that pretty recently, but also briefly talk about changes in demography in Chinook salmon coastwide, as well as other species of Pacific salmon. So Bristol Bay in southwestern Alaska consists of several highly productive sockeye salmon systems, as you probably know. with a commercial fishery that operates in Bristol Bay that accounts on average for about 50% of the global catch of this species. The plot on the left here shows changes in H composition of the return over time, where H is designated as freshwater.ocean. So 1.2 fish would have spent 1 year in freshwater and 2 years in the ocean. And as you can see, there's been changes in the H composition over time. So for instance, increasing proportions of 1.3s, decreasing proportion of 2.2s, and that's only at the bay-wide level. There's much more variation if you look at the river level. But we're particularly interested to understand whether changes in freshwater and ocean residents were associated with climate and or competition. And so first, what we found was that the proportion of fish that migrated to sea after 1 year in freshwater, as opposed to 2, so earlier, which is shown on the right here, increased over time. And that shift was associated with an increase in lake temperature or warming of the freshwater environment, which is shown on the left here, with, as you can see, pretty strongly significant and positive effect sizes. So these results suggest that there's shorter freshwater residents due to warming of the nursery lakes. And then second, we found that the proportion of fish that then following that spent 3 years in the ocean as opposed to 2 also increased over time. And that shift was associated with that early outward migration to sea that I just showed, but also with an increase in the salmon biomass in the North Pacific Ocean, which is shown on the left here, which we just saw is dominated by pink and chum salmon. And that suggested intraspecific competition at sea contributes to longer ocean residence. That negative effect of competition was also indicated when we looked at the weight of the returning fish. So this is ocean 2 fish as a function of salmon biomass is sort of tends to decline. And we therefore wanted to sort of dig into that part more and understand drivers of changes in sockeye salmon body size as well, specifically size at H, in addition to these changes in H structure. So specifically, we looked at size at age of the fish as shown on the plot here, where we have 4 dominant age groups returning. And we see pretty synchronous changes in size at age across these age groups, right? With some sort of rather stable, slightly increasing period, and then a long sort of decline in size at age. And what we did is we calculated the individual anomalies of size at age for each age group, and then averaged those anomalies across age groups for each year to get what we call the mean annual size at age anomaly, which is that time series in the center here. So this is basically a way to simplify sort of a multivariate problem to a univariate problem or time series that we can then more easily model as a function of covariates related to climate and competition. And we did this by fitting a model to the bay-wide changes in mean size at age anomaly. As I said before, with the age structure, there's much more complexity in sort of the river-specific responses, which we looked at as well, but I'm not going to talk about for this presentation. And what we found was that size at age was negatively associated with both the run size of Bristol Bay sockeye salmon and with pink salmon abundance in the ocean, sort of non-linearly. And these two sort of covariate time series alone explain about 50% of the variance in that size at age anomaly. Secondly, size at age was also negatively associated with, slightly negatively associated with summer temperature in the Bering, and then positively associated with winter temperature. And that overall suggests negative effects of competition at sea and sort of season-specific temperature effects affecting size at age. Okay, we then also wanted to know what are the contributions of shifts in size at age versus age structure to changes in mean body size. So basically putting these two together to look at changes in overall size. And what we saw is that mean body size comparing sort of the first 5 years, last 5 years of the time series, sort of a simple approach, declined by about 3%, 19 millimeters, where size at age declined by about 16, and the remainder was attributable to shifting age structure, such that shifts in size at age were the main contributor to changes in mean body size. So I also wanted to briefly compare this to size declines in other species here for Chinook salmon. And this plot here shows a model estimated mean size trend based on 85 populations along the coast, shown on this map here, for both wild and hatchery fish. And it showed a rather clear pattern of declining mean size in these populations of at least 70 millimeters, maybe more, or around 9%. Not all populations are declining in size, and there are some differences between regions, as well as wild versus hatchery fish, spring runs versus fall runs, and individual populations. But the average trend is rather consistent, a declining trend in mean size since somewhere in the early 1970s. And in the interest of time, I won't go into sort of causes of declines here, whether that's climate, competition, predation, fishing. It's probably a whole nother seminar to talk about. But the conclusion here is that there's much stronger size declines in Chinook compared to sockeye, and also compared to other species of Pacific salmon. One study that looked at that was a study by Chris Oak et al. Looking at changes in body size of Chinook, chum, coho, and sockeye. And they also found strongest declines in Chinook, about 8%, so very similar to that estimate I just showed on the previous slide. And the other species with changes around 2 to 3%. They also looked at climate and competition drivers of size declines and found that some effects of both, but also lots of variation in the magnitude and direction of these links, which I'm showing here. I don't want to talk through in detail, but it's basically showing various different drivers on the y-axis here, and then 4 species: Chinook, chum, sockeye, and coho. But I highlighted one, which is the nearshore summer SST. So nearshore summer SST was positively associated with body size in Chinook and coho, but negatively associated with body size in chum and sockeye salmon. And that suggests that both the size trends themselves, as well as the sort of effects of climate and competition can vary by species. Okay, let's shift gear and talk about climate and competition effects on productivity. Brendan gave a really nice overview already, and I'll touch on some of the things that he mentioned before. I want to briefly sort of present a couple of case studies that I've worked on over the past few years. One is on pink salmon in Prince William Sound, Alaska, and the other one is on coho salmon here on the Washington coast. So the first study I want to mention is looking at populations of coho salmon on the outer Washington coast on the Olympic Peninsula. So it's the Quileute, Hoh, Queets, and Quinault river systems that mostly drain the Olympic National Park. So pretty pristine, relatively pristine watersheds, at least for down here. And to do that, to look at sort of or assess potential effects of river flow on freshwater productivity, which is what we're interested here, we constructed a pretty simple 2-stage life cycle model where we have 2 sort of transitions, adult to smolt and smolt to adult, with marine survival and harvest. And on the freshwater side of things, we have a density-dependent sort of transition and model potential effects of covariates, in this case, different metrics of river flow. And what we found was that minimum river flow, which is typically occurs in August, September, or October, had the strongest and most significant effect. So higher summer flows increase adult to smolt productivity. So on the left here, I'm showing the time series for 3 of the river systems of the minimum discharge in summer, or throughout the year actually, which is typically in late summer. And then one case or one river system effect size and the resulting estimated smolt to adult productivity. And in addition to finding those effects based on past data, we did some simulations of expected flows in the future under current climate projections, which indicated that these populations could experience declines in freshwater productivity of close to 10%. And that's only considering flows during juvenile rearing and not adult migration. So evidence that extreme low flows decrease freshwater productivity. This is, of course, particularly relevant in this year as we just experienced some of the most extreme low flow conditions on record in the Pacific Northwest. Second study I'll mention is on population productivity of pink salmon, specifically in Prince William Sound, Alaska, where again, we were interested in effects of climate and competition, and particularly potential competition with hatchery pinks. So shown here is the time series of recruits per spawner and the natural logarithm of recruits per spawner, which is our metric of population productivity. And we specifically looked at these populations because Prince William Sound has a large hatchery program for pink salmon. And this map shows the proportions of hatchery origin fish returning to each region throughout the North Pacific, where we can see that salmon returns to Prince William Sound, which is that pie chart, the mostly yellow pie chart up there, is dominated by hatchery fish. And it's the only region where that is actually the case. Now I want to focus on two findings from this paper, among others. So first, we found evidence for climate impacts, specifically positive relationship between SST and productivity, though that relationship switched from significantly positive to non-significant, only slightly positive after that 1988-89 regime shift. And Brendan talked about that before. This has been found for other species as well in that region. The second is that the number of hatchery origin fish released into Prince William Sound, which is that purple time series on the left here, that were released in the year of juvenile migration, was negatively associated with wild pink salmon productivity, suggesting interactions of hatchery and wild fish probably in the nearshore marine environment. So evidence for negative effects of hatchery production and non-stationary climate effects. Other studies have looked at that effects of competition and sea surface temperature with pink salmon over larger spatial scales. Brendan presented that for sockeye. I wanted to sort of reiterate that using a study by Franz Müther and others. And they looked at SST effects on pink chum and sockeye productivity, showing generally negative SST effects in southern populations, positive effects in northern populations, with sort of a transition or breakpoint somewhere in Southeast Alaska or northern BC. So large-scale variation in the magnitude and direction of climate effects. Okay, so with that, I'll briefly try to summarize these findings in three main conclusions or take-homes. So first, climate and competition affect population productivity, but also life history traits, and we've seen that sort of in the changes in age at smolting and age at maturation in sockeye salmon, as well as demographic structure. So for instance, declines in salmon size that I presented. Second, salmon growth and survival at sea are impacted by competition within species. So for instance, that hatchery wild interaction in pink salmon and among species. For instance, the effect we saw pink salmon abundance on sockeye salmon body size. And then third, effects of climate can vary by life stage, like freshwater versus ocean, by season, where we saw differential effects of summer and winter SSTs on sockeye salmon size, by latitude, just presented on that. So SST effects tend to be negative in the south, but positive in the north, can vary by species, like we saw for the nearshore SST effect in Alaska salmon, and it may be non-stationary. So the temperature effect sort of pre and post regime shift in the pink salmon case. So lots of complexity here, especially with regard to climate impacts. And it's of course important to consider that when making decisions about salmon conservation and management. And at the end of the talk, I sort of want to zoom out and talk more broadly about those sort of challenges in salmon research and conservation and management. There's a joint Outlook slide that Brendan and I put together, which we thought might also be useful for the Q&A session. And we wanted to provide this Outlook primarily by asking a few questions and we've identified sort of four major challenges. I'm sure there's more. So a first challenge, limited inference about ocean ecology of salmon. And Brendan talked about that a little bit. So how do we best manage based on incomplete information? right? A lot of the things that we talked about today are based on correlations, right? Statistical correlations. They're not experimental studies. They're not process-based studies. So how do we manage, or how can we best manage based on that incomplete evidence? And then the second, as Brendan mentioned, are experimental manipulations at the basin-wide scale possible. So for instance, changing hatch reproduction. Challenge number 2, promoting climate resilience of salmon populations. So we didn't talk about that much today, but both Brendan and I have been sort of involved in work looking at the effects of biological diversity on stability in salmon systems. The question here is which management strategies or conservation actions are most likely to promote biological diversity of stocks, life histories, genetics, etc. And then given climate change, where is recovery of listed or at-risk populations likely to be possible over the next few decades? It could be 10 years, 20 years, 50 years, whatever our reference timeframe is. Third challenge is monitoring and managing populations for ecosystem change. So the question would be, how can we build flexible monitoring and management systems that can detect and respond to the ongoing ecosystem changes we observe? And then the last one, sort of related to that, is uncertainty in our understanding, the population models we use, and future projections. So how certain are projections of unobserved ecosystem states? Right. So whatever we observe, whatever we make inferences on, is based on past observations, but likely what we're going to see is states of the ecosystem that we haven't observed before. So thinking about extreme events, tipping points, non-linearities, or so-called no analog futures. And what are the most robust strategies for assessing and managing populations, given the large knowledge gaps and limitations in predicting the future? So it's slightly different than asking, given my prediction of the future, how should I be managing, right? It's sort of asking, how do we best manage given that uncertainty? Okay, with that, I would like to thank everyone for the attention, and I'll be happy to answer questions during the Q&A. Thanks.
Well, thanks very much for your presentation, Jan. Certainly thought-provoking. If you could leave this last slide up on the screen, we'll take a 5-minute break. It's time to start the moderated question and answer session, and I'll turn this over to Lori Wykamp and Marissa Litz, who are both members of the Coho Technical Committee and are actually doing a lot of the hard work in terms of organizing this seminar series. So take it away, Lori and Marissa.
So our first question comes from Gary Morishima, and it's targeted towards Brendan. He said, can you provide the references indicated in your slides? And what do we need to get inside the black box of the ocean to understand how we can address uncertainty and increase the resilience of local salmon populations, particularly Chinook, Coho, and Steelhead, originating in the southern portions of their ranges?
Okay, well, great question, Gary. Lots to unpack there. I'll start with the easiest part, which is, yes, I can provide the references. I just sent Johnson a PDF of the presentation. So I think he has both of these presentations and we'll be posting them shortly. So they'll be on there. Next part of the question, when it comes to getting inside the black box of the ocean, I feel a little bit sheepish, to be totally honest, talking about this while I'm looking up at Lori and Marissa on the screen, but I'll take a stab. I do really think, given the work that we've talked about here today, that some of the biggest uncertainties we have around mechanisms and understanding the nuance of some of these broad scale correlative relationships that we have are in part a function of our lack of understanding of the spatial and temporal distribution of different salmon stocks throughout the North Pacific. And we really have a limited understanding of where fish are when and how they're interacting, how that changes in space and time. A lot of our information at the really broad scale is from back before I was even born, when there were half as many salmon in the North Pacific as there are now. And there, I think, as everyone knows, and probably have seen presentations on the IYS surveys and the coordinated surveys across the North Pacific, while of course they don't come at, they come at significant expense, I do think that coordinated, collaborative research across salmon nations that leverages some of the amazing changes in technology that can be harnessed. I'm thinking about things like eDNA and autonomous gliders can really, really push the envelope of our understanding of where salmon are, when, and how they might be interacting. And then as a result, that can help us better understand both wild and hatchery distribution, how that shifts in space and time, and in response to kind of warming and competitive interactions. That's at a very broad scale, though. You also highlight here, Gary, in your question about what we can do to increase the resilience of local salmon populations, particularly Chinook, coho, and steelhead at the southern portions of their range. So two thoughts here that I'll share. One is, I think everybody heard this pretty loud and clear with the last set of presentations that were done in this seminar series about the importance of protecting, enhancing, and maintaining heterogeneous dynamic habitats and protecting the processes that generate those dynamic landscapes. Of course, that's a lot easier in some places than others, but that coupled with ensuring that the genetic heterogeneity that's part of the hallmark of Pacific salmon is protected are things that I think are going to be really important to helping strengthen, increase the resilience of local salmon populations. But I have to be honest, I think we also have to be realistic about prospects for some salmon populations and some region. And I know that's not a popular thing to think about. And salmon, at the end of the day, we as people are fundamentally so place-based tied to salmon. But I think we do also have to be honest with ourselves about what the prospects look like for some populations in some places, and think about some of the tough calls that we might have to make about where and when we invest limited resources to try and promote resilience and adaptation moving forward. So hopefully that helps a little bit, Gary.
Thanks, Brendan. So Gary Morishima always asks wonderful questions. So now we'll switch to Jan. So from Gary, how is environmental change affecting the food web? So in other words, alteration of primary production, prey species, and impacts to salmon. Things like anchovy and thymine and Chinook salmon fitness. And the second question, what are prospects for impacts of environmental change regarding predation on juvenile and adult salmon by pinnipeds?
Yeah, thanks. Those are really good questions. I'm not sure I'm the expert on either of those, but so first, I guess, thinking about effects on the food web and lower trophic levels, I'd say that, again, it depends on where latitude ecosystem, probably as sort of a general trend is that we'll see maybe poorer condition, lower lipid, sort of zooplankton and other prey species that may propagate up the food web as sort of general lower production. But then on the other hand, in Arctic, or subarctic systems, we may see some increase in productivity with increasing temperature, at least during times of the year that we wouldn't otherwise see a lot of production. So I think it's really hard to predict. Not sure how. I mean, there's also, of course, the interaction with salmon abundance and competition. So that the competition effect is a function of the food available, which in itself is an indirect sort of climate effect. Again, I think it's going to be hard to predict what that's going to look like. For impacts of environmental change on predation, specifically thinking about pinnipeds, I would say generally they are going to be less affected by direct effects of temperature compared to fish because they're warm-blooded. But they'll be impacted a lot sort of through indirect, again, food web interactions and their prey themselves, right? So there's this feedback between sort of impacts on salmon and impacts on the predators. I think it's fair to say that in the past, changes in at least pinniped abundances have been largely affected by human exploitation and protection and recovery. So there's much less of a sort of a, I mean, there's the climate effect in there, but it's sort of overridden by those, legislative impacts, if you will. So I think that is probably also going to be the case going forward, depending on, you know, what we're doing with the recovery of pinniped populations.
Thanks, Jan. And Scott McPherson has a question for Brendan. Brendan, do you have any theories as to why Chinook have in general not exhibited latitudinal changes seen in most other salmon species? For example, their production is generally lower across the entire range.
Yeah, that's a good question, and I definitely invite Jan to chime in once I take a stab at it. You know, I don't have any. I haven't spent a ton of time, Scott, to be honest, thinking about this, but I'll throw out two hypotheses that might contribute to that pattern, because you do point it out, and one thing that I certainly didn't highlight in my presentation is the fact that across the North Pacific range, Chinook generally in recent years have had pretty poor survival, haven't been responding generally the same way that we've seen with some of the other species in terms of a warming ocean. Again, that's a very broad brush statement. Two things might be going on. One, habitat might really be filtering the types of climate-driven changes that Chinook are experiencing differently than other species. And I'm specifically thinking about freshwater migrations of returning adults who, particularly in the subarctic regions like the Western Alaska region and in the Yukon, we've seen warming river temperatures that are associated with thermal stress, and there's beginning to be some evidence of thermal carryover effects from one generation to the next associated with warming waters and long distance freshwater migrations. That might contribute to some of the depressed productivity that we see in Chinook, disproportionately relative to things like pinks or chum or sockeye that can take advantage of or benefit from changing phenology in lakes associated with warming. The other one is one that Jan highlighted. I don't think this is an either or. You know, the very striking temporal change in the size of adult fish returning to spawn, which then has consequences for the reproductive potential of those fish that do make it back to spawn. Chinook have the most striking declining patterns across their range. And you could imagine a situation, again, I'm just totally speculating, you could imagine a situation where declining size, perhaps due to predation, perhaps due to climate effects, then results in reduced reproductive output that's compounded by freshwater migration stress to be a bit more of a perfect storm relative to other species that don't tend to have the same large changes in demographic characteristics and don't necessarily have the same long distance freshwater migrations. I'm sure there's probably all sorts of holes you could poke in those general ideas, but those are a couple to chew on a little bit.
Thank you. So next we have a question for Jan, and this is a really general question from Dan Auerbach. How can salmon harvest management best accommodate evolutionary or genotypic adaptive potential? So I'm thinking of demographics and some of the other trends you talked about.
Yeah, that's a really good question. I guess there's different parts in here that I'll try to talk about. So one is sort of the evolutionary, one is sort of the potential question. How much potential is there that could, you know, compensate for potential negative effects? I would say there is, you know, current projections for climate change are that it's going to be so rapid that there's actually going to be a little potential, adaptive potential for short-term evolutionary changes to sort of compensate for that potential negative effect in terms of their thermal physiology, et cetera. And if there is, I guess from a conservation perspective, it's not too worrisome because it would mean that, you know, there's some compensation going on with respect to the effects that we would predict otherwise without that adaptive potential. The other aspect is harvest management responding to long-term changes in size, age, and those sort of demographic traits, which likely is partially an evolutionary response, but we don't really know how much of that or on what time scale. But the harvest management, by accounting for those changes and estimating, and sort of, or building that into underlying models that inform harvest management, could, accommodate that. It's possible to think of, typical spawner recruit analyses that inform, reference point estimates that are used in management to account for that change by directly sort of, instead of using a spawner as the sort of reproductive unit, directly translating it to reproductive potential or fecundity or egg mass, things like that, and sort of building models based on that.
Thanks, Jan. Our next question for Brendan is from Rosie Gradeville. She says, what do you think likely drives the correlations with sea surface temperature? Are they direct effects of sea surface temperature on salmon, ecosystem effects linked to climate, primary production, prey availability, etc., or is it something else?
Yeah, great question, Rosie. Highly unlikely to be direct temperature effects on salmon. The temperatures that we're talking about are still well within the range that generally Pacific salmon can respond to. So the general thinking is that it's, a coarse index of production and ecosystem state that, like a classic example is copepod community structure as a function of temperature. And you tend to have more lipid-rich copepods in relatively cooler waters than you do in relatively warmer waters. And that then translates into energy transfer to juvenile salmon that are feeding on them, et cetera. So the general idea is that absolutely SST is just a convenient index, if you will, of broader ecosystem changes that co-vary with temperature, probably in all sorts of hard to tease apart ways, that then are what are truly ultimately influencing the way in which temperature propagates effects up through the food webs, and then also probably then also how it influences other competitors or predators as well at different life stages while salmon are at sea. But I will say, sorry, this last thing I will say, because a lot of the temperature correlations that we talked about in our presentations were temperature just before or during early marine life, it's more likely to be that this is kind of a combination of ecosystem-y type effects, food web type effects during that kind of early marine period, as opposed to what's going on later in life at sea.
Thank you. So our next question is from Bill Bosch, and it's for Jan. So we have observed younger age at return, so about a 8% decrease, which would correlate to smaller size. This is the Columbia Basin spring Chinook hatchery population over a 13-year study period. So again, brood years 2003 to 2014. So we correlated this with a larger size at release for those broods over time. They haven't had yet time to investigate whether feeding regimes, rearing, water temps, or other factors might be causal. So his question, I'm wondering if warming river waters might be, at least in part, be the cause of younger age at return. Do we know if any wild Pacific salmon population are showing larger juvenile smolts or migration size over time?
Yeah, it's also a great question. I'll start rolling it out from the back here. So I don't know much about time series or sort of changes over time and smolt migration size. I can say that when we look at the sizes of fish that return for the younger ones, and that is true especially or both for wild and hatchery, we don't see a decline in size at age for fish that return after only 1 year in the ocean. So which might suggest that, there is not much change. If anything, we see a slight increase in that size at age. But if we go, if we look at H1, H2, pretty stable. H3 starts declining over time. H4 strongly declining, the same with H5. So it looks like the growth environment early on is not sort of causing smaller size at age. Yeah, so and I don't have anything to pull up for juvenile small migration size. But the other question, I'm wondering if warmer river waters might be part of the cause. Sure, it could be. There is, that general sort of life history response to increasing rearing temperatures is a faster growth, but an earlier maturation at smaller size and younger age, right? So that could be part of that warming response. However, we see those declines for size at age and mean size throughout the range pretty much. So that's sort of a counter argument for that being a major driver of at least sort of the general trends that we see for Chinook. But thinking about the Columbia River, it certainly could be part of it because again, it's relatively far south, so temperatures are reaching or have reached close to or above optimal temperatures in many years. So I think that can be part of it.
Great, thank you, Jan. Question for Brandon from Joy Thorkelson. And I'm not entirely sure what the question is, but I think just generally speaking to the fact that DFO policy has had a decrease in harvest rates since the 1990s, but at the same time, recent data from Rougeroni and Irvin showed that the biomass of fish or numbers of fish was relatively constant, except for a 10-year period in the mid 1980s to early 90s. And wondering if you know what the causes of that increase were. Is this high productivity with hatchery releases, or is there something else going on? And then she also asked in a follow-up question about the pre-contact numbers of salmon in the North Pacific, which I think is also a very interesting question, if you have any insight into that.
Okay, great questions. I'll try to touch upon them to the best of my abilities. I think, Joy, you're right. If I'm reading correctly here, I do think that there was a fairly extended period through to the mid 80s and into the early 90s with relatively stable commercial harvests in BC that then really ramped up in the 80s and 90s and then have really tailed off. And they're definitely almost certainly influenced by environmental conditions, but also all the other things that are going on that regulate harvest, including mixed stock fishery challenges that have become more acute in recent years that have resulted in reductions in harvest, particularly marine mixed stock harvest. Hatchery production, at least in BC, is not as large as it is in other regions. And so I would be wary of ascribing a lot of the big ups and downs in BC specifically to hatchery production. But then on the question around, wondering what the numbers of fish look like pre-contact, great question. To my knowledge, the best evidence we have of long, like decadal, century scale, millennial scale changes in salmon production come from limnological cores in sockeye lakes. There's been some really fascinating work that's been done that's been able to go back and look at changes in marine-derived nitrogen, I think, probably put my foot in my mouth, in those lakes over very long time scales that can then be calibrated for periods of contemporary estimates of absolute abundance in those lakes. And they show extremely large, centennial scale ups and downs, which really, remind us of the dynamic world that we live in. That's not really a big surprise, but that, we do really end up with our blinders of our frame of reference of the period that we've worked with a system or, interacted with a system, derived benefits from a system. That doesn't give you a great answer of what, say the absolute production was pre-contact. The slides we were showing today only go back to the, turn of the century, which is still quite a time period, but by no means that far back when you really start thinking about things. But yeah, it's kind of what I got on that question for right now.
Thanks, Brendan. So you're not entirely off the hook. I'm going to pose this last question to both you and Jan, and if Jan wants to go first. But Brendan, you presented the controversial notion of reducing hatchery production. So Gary's asking, can you provide some examples of the types of basin-wide manipulations involving hatchery practices that could be, implemented, or provide some ideas about how they might be evaluated? So Jan, if you want to go first, and then Brendan.
Sure, can give it a try. Yeah, of course, that's a really complex issue with legislative, economic issues or consequences would require international negotiations and so forth to change hatchery production at a large scale. But it's one idea, and as Brandon said, one option for us to control sort of the ocean environment where we don't have many others. So this could be done sort of multiple years in a row with either up or down regulation. In fact, I think both might make sense from an experimental standpoint. Would have to be probably at least a life or ocean residence time of the salmon species that we're interested in. So probably somewhere between 2 and 4 years. And if I would imagine it, I could imagine some, down regulation for 3 years, up regulation for 3 years, and after that assessment of the consequences of that. But I'm of course aware that is requires a lot of international effort, and not sure this year is the best to do that. Brendan?
I'll just throw in a couple of extra thoughts. I mean, I think Jan is bang on. It would be extremely complicated to think about whole basin scale manipulation of hatchery production, and in some ways might be almost intractable from a kind of resource perspective to truly monitor responses. But one of the beauties of a fixed 2-year life cycle with pink salmon, which of course is only one of the species, but the most abundant species that we have out there, is that you get extremely large changes in abundance from 1 year to the next. And we didn't talk about it today, but part of the large body of evidence of kind of ecosystem-y responses to pink salmon come in looking at responses and different ecosystem characteristics at different spatial and temporal scales to this high frequency variation in pink salmon abundance. And so we see this in plankton, in other fishes. We see it in breeding success in marine birds, possibly even in southern resident killer whales in terms of their ability to forage. So there are natural elements to the fixed life cycle of pink salmon that help shed some of the light, and I would argue have contributed to some of our growing body of evidence understanding of competition effects at sea. The other thing that we could think about is manipulation of hatchery production at smaller spatial scales that might be more tractable within individual nations. Jan walked through a really excellent example of trying to tease apart interactions within Prince William Sound, where you have very large hatchery production, like large magnitude hatchery production. That's a system that falls wholly within a single nation that could, in theory, manipulate production if that was something that was desired to shed light. But closer to home, you could imagine a situation like in the Salish Sea. But we already see in the Salish Sea extremely strong biannual variation in pink salmon abundance, at least, which has helped provide. I mean, Marissa has done some great work on chum salmon. There's lots of work that's looked at interactions at that kind of finer scale. So there are other scales at which we can try and get at some of these questions. One of the things I posed as a bit of a stirring the pot type question at the IOIS symposium a few weeks back was whether or not one of the challenges with large scale manipulation is who's going to pay for it, and then how do you compensate those countries that maybe reduce production? Then how are you going to pay for going out and surveying at sea and trying to understand the ecosystem? Well, one way you could think about it is maybe levying a small tax on hatchery releases over a period of time that would build up fairly quickly, with 5 billion fish being released fairly quickly, a bit of a kitty, a research type kitty that could help offset some of those costs and also help support some of the targeted research. Again, just throwing it out there, and you heard it here first. Jan was the one who was suggesting that we do this, not me. I'm just joking. But I think like the reality is that large scale experimental manipulation is the gold standard when it comes to understanding cause and effect. And as people spend lots of time thinking about trying to understand relationships and associations, you're ultimately, your hands are a bit tied without real manipulation of ecosystems, and it gets harder and harder to do it as the scale gets larger and larger. But I think that there's a lot of room and space for squeezing more insights out of some of the unique quirks and nuances of life histories with some more focused and targeted research.
Thank you, Brendan. That's a perfect lead in to our November seminar. And so we have run out of time. It's almost 3:30, which is usually the end of our Q&A session. November 30th will be the next seminar, and it's actually about hatchery practices. So we've got the Bryans. Brian Riddle from the Pacific Salmon Foundation is going to talk about some of their recent research on hatchery practices. And then Brian Beckman with the CSC is also going to talk. And he has a really interesting, perfect example of this is, you know, what are we doing with our hatchery programs and what are we trying to achieve? And I should also point out that Oregon Coast Coho, they completely or essentially closed down all hatchery releases because of a big stick, namely the Endangered Species Act, and have seen a pretty impressive increase in coho returns to the Oregon coast. So there are some examples that are interesting. And with that, I hope to see you all on November 30th for the next installment of this seminar. And thank you again to our two speakers, Jan and Brendan. This has been really fantastic. So with that, I'll sign out.
Thanks very much. Good to
Thanks,
see everyone.
everyone.